Our topic is DNA and protein nanostructuring. In part 1, the protein carries in DNA structure. DNA is very good for structuring, but the chemical functionality is not so good. So we want to combine it with proteins. For that, we created DNA origami. DNA origami is a nanoscale folding of DNA to create arbitrary 2- and 3-dimensional shapes. Folding along single strands of phage DNA named scaffold strand aided with smaller single strands, the staple strand provides a DNA origami after heating and cooling. On one hand, we produce the DNA origami with a triangle design like this. And modify them at one edge, recognized by bivalent binding antibodies. This enables us to link the origamis to dimers. If we use multiple chemical modifications, we can get higher order oligomers. On the other hand, we modify the triangle with an alkene oligonucleotide at one edge. And add a peptide. It has an aza group at the N-terminus and an LPXTG motif at the C-terminus. Via click chemie, we can couple directly. It is a chemical coupling. And this is the result. To combine the encoding properties of DNA with the recognition and catalytic functions of properties, we use the short amino acid sequence which is recognized by the enzyme sortase. This protein can catalyze the ligations of peptide, DNA origami, and the protein containing also the sortase motif.
In comparison to the first part, where the protein is displayed on the DNA structure, we use protein structure, to deliver DNA. In nature exists a model to deliver DNA, and it is called virus. Into the viral capsid, we introduced recombinant proteins. And use the virus for the transfer of DNA between virus and the cells. The adeno-associated virus is a small non-pathogenic virus with a moderate immune response in the host organism. To reduce its broad host range and to target the virus against specific receptors, recombinant adeno-associated virus with altered surface are produced. The production of recombinant adeno-associated virus proceeds in human embryonic kidney 293 cells. By co-transfecting with 4 plasmids. The first one encodes targeted ligand, which is fused to the viral capsid. The other one encodes gene of interest, flanked by ITRs, which is then packaged into the viral capsid. The other 2 plasmids are required for the viral assembly. Only a few of the 60 subunits of recombinant adeno-associated virus are modified. The recombinant virus can effectively infect specific cells. Because of its efficient and specific delivery of genes to the cells, the recombinant adeno-associated virus has gained a big interest in gene therapy. To allow native expression of proteins or synthetic production of modified peptides, the ligation of the peptides in a hemoselective manner could be done. There are many possibilities to connect 2 peptides to each other, but we focused on the enzyme sortase because of its directed ligation. The completely expressed proteins or synthesized peptides contain the C-terminal, sortase motif, pentaglycine, which is recognized by the enzyme sortase. The N-terminal sortase motif was fused to the N-terminus and is called LPXDG. To allow the ligation by the sortase, regenerating a native amide bond. Such a method allows us to fuse several peptides to the surface of the virus and maybe use it in medical researches. In our project, we use DNA structure to display proteins and highly folded protein structure to deliver DNA. There are also many methods to connect DNA with proteins, but we focused on click chemistry and sortase because of its directed ligation.